Oh, the essay brunch is 10 a.m. on Friday. Okay, I'm going to turn it over to Jim McGuire, who's going to introduce today's speaker.
All righty. Well, it's my pleasure to introduce Tandy Warnow, today's speaker. This is a really impressive turnout. I warned her that there might be a fairly empty room today because the semester hasn't started yet. And this just goes to show what a great draw you are. So Tandy's here on a bit of a speaking tour. She's already have you already given your other talks at UCSF and at UC Davis. And as I'll go through her vita a little bit in a moment. You know, she did her undergrad and her PhD here at Berkeley, and she was just telling me, I said, well, why are you actually here? I didn't mean for it to be taken the wrong way. It's like, why wouldn't I come to Berkeley? Berkeley's like the best place in the world. So we all know that. Tandy knows that. Welcome back to Berkeley. So just a little bit about her history. I've already told you that she did her undergrad and PhD here at Berkeley. She did a couple of postdocs. She did a postdoc at USC, and then she also did a postdoc at the Sandia National Laboratories in New Mexico. That's New Mexico, right? And then she had her first faculty position at the University of Pennsylvania. She was there from 93 to 99, and then she moved to the University of Texas right about the time I was leaving Texas, getting my PhD. And she was at Texas from 1999 until 2014, and she's just moved to the University of Illinois. At the University of Illinois, she's in the Department of Bioengineering and also in the Department of Computer Science, but I noticed on your vita that you were also linked to about 10 other departments, so it seems like it's a big deal. Tandy, of course, is very well known for her work developing especially algorithms for phylogenomic analysis, also for multiple alignments, multiple sequence alignments, and also for metagenomics. And in fact, I noticed that you have NSF grants now for each of those three topics. And another grant as well. There was a fourth grant as well. So obviously she's still developing, new algorithms and such that are useful for all of us in all those areas. And I'm really happy to have her here to give this talk today. She's going to talk about new methods for estimating species trees, that part of her program, from genome scale data. And there are a number of people here in the museum and in the department who are really interested in this problem. And one of the methodologies that she's developed at Astral is a program that a number of us have gleefully utilized for our next-gen data sets that we've collected for phylogenetic analysis. So it'll be great to hear more about those methods. So thank you for being here, and the floor is yours.
Thank you. Okay, yeah, I really do love Berkeley. It is really the best place. So what can you do? Okay, so I don't need to give a lot of background, but just for the context, we're going to be talking about species tree estimation and doing this by sampling multiple genes from throughout the genome. And I'm going to be talking about the problem of gene tree heterogeneity, and I'm hoping that most of you know about this because I'm not going to be going into this in detail. But when you sample genes from throughout the genome, some genes actually have different trees from others. And so it becomes an issue about how are you going to get the species tree. You can't just pick one gene, you have to look at several, and then you have to figure out how to address different reasons for gene trees to be different and use that information in a rigorous way. So phylogenomics has at least two meanings. Jonathan Eisen coined one version of the term. The term that we're going to be using means how do you do genome scale phylogeny, so phylogenomics. And I'll refer to genes. They're not really genes. They're just different regions of the genome that are supposed to have no recombination within that region. Okay? So how do you use different regions of the genome? We're going to just call them genes. And the trees on these regions are going to be called gene trees. How do you use this to get the species tree? I just want to point out that the kind of trees that we're reconstructing are unrooted trees. So gene tree discordance can be due to multiple causes. Incomplete lineage sorting is one of the causes. It's modeled by the multi-species coalescent, and it's considered perhaps the dominant reason for discordance between gene trees. I'm not going to address whether or not it is the dominant reason, but just talk about methods for estimating species trees when discordance is due to ILS as opposed to anything else. So the multi-species coalescent is a backwards process, but it's equivalent to a forwards process. And basically what it's saying is that you're modeling populations. So every dot is an individual, and you're tracing history back in time. And so what you have in a situation like this is that as you trace the evolution back between these two individuals for these alleles, they actually coalesce in the lineage that they could, so in the branch, the first branch that they could coalesce in. But when you look at these two branches, they do not coalesce in that branch. And so that causes the potential to have a tree that's different from the species tree. And in particular, this gene tree that's inside this species tree does not match the species tree. So in the gene tree, you actually think that these two are sisters, and they're not. So the things that increase the chance of this discordance is very short time periods for speciation events, right? So if you have a very short time period, you're more likely to miss that coalescence, and population size also affects it. So these coalescent branch units, which are a function of these two parameters, impact the probability of having a discordant gene tree. So when you're trying to estimate a species tree, if you have a rapid radiation, which is to say very quick succession of speciation events, you're more likely to have this kind of discordance. So this problem came up in two projects that I was in. One is the 1,000 Plant Transcriptome Project. We published our first paper in 2014, but the next paper will involve a much larger data set. But even in this first paper, we had massive gene tree incongruence that we thought was consistent with a rapid radiation causing incomplete lineage sorting. So we had to deal with that issue. And in the avian phylogenomics project, we also had massive gene tree discordance consistent with ILS that we had to address. It turned out, even though both of them were published at approximately the same time, which is late 2014, we actually had different methods that we used for these two analyses, simply because the methods were developed in order to address these problems, and the methods were developed at different times. And Astral, which you just heard about, was one of the methods we developed to be able to deal with this. So this talk is going to be about the methods that we developed. And in particular, I'm going to tell you about Astral, and then I'm going to tell you about statistical binning, and then I'm going to tell you about some controversies involving statistical binning and what's going on there. And I would actually welcome questions during the talk, and I'll try to make sure that I complete on time. So if I can't answer your question during the talk, and I'll say that I'll answer it afterwards. Okay, so statistical binning is controversial, and I'll be telling you what the controversy is. Okay, incomplete lineage sorting, it confounds the analyses of many groups, and there is a big debate in the community about how to do this kind of analysis, how to do a phylogenomic analysis in the presence of incomplete lineage sorting. And it's really focused on the statistical guarantees, which is to say theory, and yet performance on data. So there's sort of a conflict between these two perspectives. So statistical consistency, most of you know what this means? Yes? No? Raise your hands if you do. Okay, half. It's as the amount of data increases, your error should go down. It doesn't have to go down monotonically. But basically, you should be able to get low error with high probability. So however much error you're willing to tolerate with whatever level of probability, you just have to have a certain amount of data to achieve that. So statistical consistency says that your error should go down, get as small as you want it to be, with as high probability as you want, with enough data. So what is data in the context of phylogenomics? It could be the number of sites that you're looking at, it could be the number of genes that you're looking at, whatever, but it's the amount of data. So the two competing approaches for phylogeny, the two main competing approaches, because these are not the only two, but the two main ones, are when you have multiple genes, you can estimate trees on each of those loci, and then you can combine the trees. So I'm going to refer to that as a summary method. There are lots of examples of summary methods in use, and some of them are designed to address incomplete lineage sorting. The other approach is just concatenation. Just take all of your alignments, put them into one big super alignment, and then analyze that super alignment. How you analyze the super alignment could be maximal likelihood, it could be a Bayesian approach, but you're just concatenating everything and running it through your software. Those are the two main approaches. So what do we know about these? The traditional approach of concatenation, it's extremely common. For example, if you look at our avian paper, we presented a concatenation analysis. That was the tree that we used to answer questions about bird evolution. If you look at the plant paper, we presented a concatenation analysis. that was the tree that we used. If you look at the insect paper that was published about a year and a half ago, that was what they did, right? Everyone's doing concatenation analyses. It's the dominant way that people do species trees. And yet it can be statistically inconsistent and even positively misleading. The difference between that is that positively misleading says as the amount of data increases, not only do you not necessarily converge to what you want to converge to, but you might converge to something else. And that's worse, as opposed to simply not knowing what you're getting. And the proof of that was published very recently by Sebastian Rock and Mike Steele. Sebastian Rock is also a Berkeley graduate, I will point out. And there's mixed accuracy in simulations. So, by the way, the proof for statistical inconsistency for concatenation is for an unpartitioned maximum likelihood analysis. So a partitioned maximum likelihood analysis is different. Unpartitioned means you just put them all into one matrix and treat every site as being IID down the same tree with the same branch lengths. Okay? That's the unpartitioned analysis. That's where the theorem has been proven. Okay, so mixed accuracy in simulations, but there is a theorem that says it can be inconsistent. The mixed performance in simulations is really interesting. There are conditions under which concatenation outperforms all the current methods. And those conditions are basically not too much ILS, not too much gene tree heterogeneity. And then there are conditions where concatenation is worse. And those are generally conditions when there's a lot of ILS. So the question about whether or not concatenation is a good idea or a bad idea really depends upon your data. Theoretically, it can give you the wrong result. Empirically, it depends. Okay? So that's the debate. Now, what about summary methods where you combine gene trees? How should we combine gene trees? We have, let's say, 10 taxa, and we have 1,000 genes, so we have 1,000 gene trees. How do we use those 1,000 gene trees? Should we just take the most frequently observed gene tree? That's one way. Turns out that doesn't work. Okay? This is called the Anomaly Zone. You can have situations where the most probable gene tree is actually not the species tree. So you do not want to just look at all your genes and say, whatever gene tree shows up the most often, that's what I'm going to put my money on. Consensus tree, like the majority consensus, it's not going to be statistically consistent. How about an MRP super tree? Not going to be consistent. So almost all of the things you might think to do don't work, in the theoretical sense of having no consistency guarantee. On the other hand, there are statistically consistent methods. MPS is one that works on rooted gene trees and will give you a correct species tree if you have enough genes as they go to infinity. The proof of consistency requires that you correctly root all the gene trees, which holds when you have a strict molecular clock and may not hold otherwise. NJS is a method that doesn't require a strict molecular clock, but it will give you a guarantee of getting the correct tree. Probably none of you have used NJS. It's not actually very well known. But there are others. The population tree in Bucky, Astral, which you have heard about, Astrid, and others are statistically consistent ways of summarizing the gene trees in such a way that you're guaranteed to converge to the true species tree as the amount of data goes to infinity. So statistically consistent. In the plant transcriptome project, we had massive gene tree heterogeneity. We could not use MPS because we could not root the gene trees. And the reason we couldn't root the gene trees is we had too much missing data. So if you're always using an outgroup to root your gene trees and you don't have an outgroup in one of your genes, you can't root it. So we couldn't use MPS. And we also couldn't use it because there's too many species. It had about 100 species, and MPS actually doesn't run efficiently with that many species. So we developed Astral, and you already heard a little bit about Astral. Astral is statistically consistent. It runs in polynomial time, and it doesn't require rooted gene trees, and therefore it's actually inherently more robust to missing data. So the idea behind Astral is extremely simple. In fact, it's so simple that you wouldn't even perhaps call this a coalescent method. So what you do is you take your gene trees, and then for every 4 species, you look at all of the gene trees on those 4 species, and then you just say, what is the frequency for each of the 3 possible 4-leaf trees on these 4 species. And you write down those frequencies, and those are like these weights on the gene trees, and then you're looking for a species tree that minimizes the distance to the gene trees using these quartet trees. So it's just for each gene tree, how many quartets does it disagree with? And you add up those distances, you want to minimize that. It's a very simple quartet-based approach that doesn't even talk about the coalescent. But theoretically, in other words to say, we can prove that it's statistically consistent because quartet trees don't have anomaly zones. So the most frequent quartet tree on every 4 species will be the species tree with probability going to 1. So, theorem, it's statistically consistent under the multi-species coalescent. If you solve this exactly, finding the provably optimal tree, it's statistically consistent. If you constrain the search, it remains statistically consistent if you constrain it appropriately. And it actually then becomes polynomial time. So the constrained version runs in polynomial time, has open source software, it's easy to use. We've been updating it frequently, so if you've been using an old version, please make sure that you're using a relatively recent version because the recent version actually handles missing data better. It was published in these places, and it was used in the PNAS paper and also used in other papers by people not involving any of us. Okay, so how well does it work? I'm going to show you some quick results. These are simulation studies with different numbers of species, different numbers of genes, different numbers of ILS, deep versus recent speciation. The software we're using for the simulation was developed by David Posada and his graduate student, Diego Malo. It's called SimPhi, and it has just been published. So SimPhi has this very nice complex model for doing this kind of gene tree evolution. And then down the genes, you evolve sequences. So we compared Astral to NJS and MPS and concatenation. And we're going to look at topological error. So here's the first result. This is what happens as you vary the number of species, but we're using 1,000 genes. We have a relatively moderate amount of ILS. And this is the error that we're showing here. So on 10 species, they have the same error, okay, which is about 1.5%. So 1.5% of the tree is missing. At 50 species, there's a big difference. Now, what are we comparing? We're comparing Astral to MPS. At 100 species, there's a big difference. Now, remember, we're looking at estimated gene trees. We are not looking at true gene trees. So what we're doing is we're taking the sequences that have evolved down the gene trees, we're computing maximum likelihood trees, and we're combining the maximum likelihood trees. And you see a big divergence in accuracy, okay? The error is really increasing for MPS, but it's not increasing very much for Astral. As we increase the number of species, MPS can't even run because we limit it to a 24-hour analysis, and Astral can. So the error climbs a little bit, but not too much. And I just want to point out that this is 1,000. So at 1,000 species and 1,000 genes, it's able to run, and it has very good accuracy. NJS is in between. So NJS was also developed by Liang Lu. It's in between the two, and it's remarkably competitive. So NJS is really quite an interesting method. We have an update on that called Astrid, which has a different way of computing trees once you have the distance matrix. Astrid is better than NJS because it's using something other than neighbor joining to compute the trees. But even so, I don't really understand why it's so good. I mean, I understand the theorem, why it's statistically consistent, but I don't understand why it works so well in practice. But it's worth looking at. Okay. What about running time? This is what happens as you increase the number of species. This is running time in terms of hours. And you can see MPS jumps, NJS increases, and Astral also increases. But Astral can actually run reasonably on large data sets. Okay, so it's relatively efficient for large data sets. Okay, biological data. This is an interesting comparison between MPS and Astral. The main thing is that there was a publication in 2014 that was basically saying, look, concatenation and MPS have a different placement for Ambarella. And the argument was the reason that it was different is concatenation is not statistically consistent under ILS, and MPS is, and therefore MPS should be considered correct, and the concatenation one should be considered incorrect. What's interesting is that Astral is actually agreeing with concatenation. So what that tells you is you can take two different statistically consistent methods and get different results. And therefore, statistical consistency by itself doesn't tell you that a method is correct. Okay? It's a much more complicated question about when is a method correct and when is it not. You have to actually look at lots of issues, and in particular, incomplete lineage sorting is not necessarily the cause for the differences between the methods. Okay, lots of groups are using Astral, so that's nice. Now onto the Avian project. The Avian project, we actually did the analysis before we developed Astral. So I'm just going to tell you, we didn't have Astral at the time we did the analysis, so what we had to do. So we had massive gene tree heterogeneity that was suggestive of ILS. We looked like a rapid radiation. We were concerned about incomplete lineage sorting. When we tried looking at MPS on this, we got a tree, in fact, that did not look anything like the concatenation analysis. It was extremely different from the concatenation analysis, and actually different from a lot of prior studies. So we were concerned about this. And we already had some evidence that MPS had problems on large data sets in terms of accuracy. But here's the killer. We had 14,000 different loci and 14,000 different gene trees, and they were all different from the concatenation analysis, and they all had low resolution. Low resolution, which means to say the average bootstrap support was 25%. So imagine 25% is your average bootstrap support. You don't have a lot of confidence in your data. Okay? And what I mean by average bootstrap support, I mean if you look at all of the edges overall, the average support was 25%. Okay, so standard coalescent methods just don't do well if you have really poor gene trees, it turns out. So they have theoretical guarantees if you have correct gene trees. They have no theoretical guarantees if you have estimation error. So that was a concern, right? So statistical consistency, this argument that as data goes to infinity, your error will go to 0, is assuming true gene trees. Okay? If you look at the proofs, there's absolutely depends upon true gene trees, no estimation error at all. So this is really the problem. Typical data have estimation error, and the theorems depend upon no estimation error. So what are you going to do? That's the debate, right? Multiple studies show that, multiple studies means lots of people, lots of labs have discovered this. It's not new anymore. That summary methods can be less accurate than concatenation when you have high gene tree estimation error. And genome scale data has lots of data, not all of which is evolving at the right rate to get correct gene trees. Some of it evolves too fast, some of it evolves too slow. And if you remove data that is giving you poor gene trees, you're potentially biasing your study. So it's not a simple question like throw out everything that you don't have good support in. That may not be theoretically the right thing to do. It might be empirically the right thing to do, but it may not be theoretically the right thing to do. So another last thing is that some have argued that in order to be consistent, statistically consistent, you actually have to also have no recombination inside your locus, and therefore you should have very, very, very short genes, right? So 10 sites. What can you do with gene trees estimated on 10 sites? It's hard, right? So these are some of the challenges that have been raised, and this is actually a lot of the reason why it's an uncertain resolution. So gene tree estimation error is the key issue in the debate, one of the key issues. So in summary, the avian phylogenomics project, most gene trees had very low bootstrap support, suggestive of gene tree estimation error. And therefore, we had to analyze our data and address ILS in a way that would work. So we had the solution that we came up with is called statistical binning. And we have a collection now of papers about this approach. The first one was published in 2014 in Science, where we demonstrate that statistical binning actually improves coalescent-based analysis. The way that it does this is that it improves the gene trees. So it gives you better gene trees, and therefore you get a better species tree. And we use statistical binning to do a coalescent-based analysis of the avian data. And the interesting thing was there, you remember I said that MPS on those gene trees was a less accurate, it was a very, very different kind of tree than we had seen from the concatenation analysis or from prior analyses. When we use statistical binning with MPS, okay? Statistical binning just gives you better gene trees, and then we used MPS. We got a tree that looked very much like the concatenation tree. And so it was an interesting consequence of using statistical binning. So, the idea behind statistical binning. The idea is, if you remember correctly, that concatenation, even though it's not statistically consistent, it actually regularly gives good results, which is to say better than alternative methods, when there's low ILS. So there's not a theorem, it's just an empirical observation. If there's low enough ILS, it can give you very good results. So that's the first thing. And the second thing is that gene tree error decreases in general as you increase the number of sites. So the gene tree estimation error goes down. This is just the statistical consistency for estimating gene trees. So the two things together suggest the approach. If you have loci that don't have enough phylogenetic signal, that you can't get good gene trees, which is what we had in the avian project, what if you could compare different loci and say, how much confidence do we have that they might actually have the same tree? And if you think that the differences in their estimated trees might just be due to random resolution around short branches, then maybe they could be put in the same bin. So you use that kind of idea to partition your genes into bins, and now on each bin you think there's not a lot of heterogeneity, there may in fact be no heterogeneity in terms of the tree. So now you can compute a new gene tree on each bin, and that can replace the old gene tree. And those gene trees become the things that you use to compute the species tree. So this is the picture. You start with your sequence data, you get gene alignments, you get estimated gene trees, and then if you're not doing binning, you just combine these trees. Instead, from these estimated gene trees, we're going to now make a graph. Every vertex here is a gene. And we have a threshold that we use to say whether or not two genes actually have different trees. And that's based upon their bootstrap support. So if they have conflicting branches with high bootstrap support, we won't put them in the same bin. And if we won't put them in the same bin, we put them in an edge between them. So we get a graph. The vertices are the genes, edges represent strongly supported conflict. And then we color the vertices of the graph so that no two vertices that are adjacent get the same color. And then we take every vertex in a given color class and put that in the same bin. So that's what we would get by taking this graph and partitioning the vertices. And now any two vertices that are in the same bin are not connected by an edge, and therefore they don't have strongly supported conflict, and therefore we can analyze them using concatenation. So this is actually an NP-hard problem. It's a hard problem to solve, but we have a good heuristic. So coloring the vertices is the hard problem, but now we have color classes, now we get new gene trees, and then we combine the gene trees. That's statistical binning. Here's the impact of using statistical binning on species tree estimation for a bunch of different methods. MPS, MDC, which is minimized deep coalescence, which was recommended by Wayne Madison years ago. MRP, the supertree method, MRL, another supertree method, and greedy consensus. This is a simulation study based upon data that CLNA developed, very heterogeneous models. This is error. Red is the method before binning, blue is the method after binning. They all get better. It's really sort of a general technique for improving species trees, but the reason it improves them is that the gene trees become more accurate.
Can I ask a quick
Yeah.
question? What does concatenation look like in this instance with these data?
I don't remember. That's a good question. I will have to look it up.
Probably poor though, right? Because this was a strong ILS.
Yeah,
data set.
strong ILS has relatively high ILS, and there's no molecular clock, and I'll have to look. But and this figure's not published. On the other hand, the comparison should be available from another paper that I can give you. Okay, so here's a theorem. I'm not going to show you the proof, but the theorem says statistical binning that I just described is not statistically consistent. So the problem is on every bin you're taking one gene tree. That's the problem. We're trying to keep these bins balanced in size, and if we were always able to keep them the same size, we'd be fine. But you could actually have bins that have different sizes, and that's a problem. So there's a theorem that we published in PLOS One that says it's inconsistent. But there's a really simple fix, which is to say you simply make as many copies of the gene trees as the number of genes. And then you completely fix the problem, and this weighted version of statistical binning is statistically consistent. So if you want to do statistical binning, just do the weighted version. So theorem also proved in the PLOS One paper, weighted statistical binning is statistically consistent under the multi-species coalescence. So you just have to remember that statistical binning and weighted statistical binning is a pipeline, right? You're binning things and you're computing trees on these bins. And then you're combining that with a method like MPS or Astral or whatever. So it's the whole pipeline we're talking about being statistically consistent. Okay, so two theorems, the proofs are published, and you should just look it up. I'm going to show you how well this worked. I'm supposed to finish in like 7 minutes. Okay, we have done fairly extensive analyses of simulated and biological data, and it's all published, and I'm just going to show you some of it. The thing to note here are these numbers. So these numbers are the average distance between true gene trees and true species tree. So the average topological distance. So a number like 84 or so means 84% topological distance between an average gene tree and the species tree. That's extremely high gene tree discordance. This is not about estimation error. This is a huge amount of ILS. So you might ask whether or not you're going to encounter this kind of ILS in your particular data. On the other hand, 18% is relatively low. It's not extremely low, but it's not anywhere close to that. So which ones of these are sort of reasonable estimates of real data depends upon your data. I think that these are extreme conditions. Okay, so we're going to show results on various things. The mammalian one is based upon a mammalian phylogeny. The avian one is avian phylogeny. So these are various levels of ILS and various numbers of taxa. Here's the first thing about the species tree topology. As you increase the sequence length per gene, and we run MPS. Here's the same thing on the same data running Astral. And what I'm mainly comparing here is unbinned methods versus binned methods, weighted or unweighted, and then concatenation. So what you see here is binned. Both binned versions are here. They start off at around 13% error, and they decrease in error to what looks like about 5 as you increase the sequence length per gene. What about unbinned? Up and down here. So there's a big gap between unbinned and binned that only decreases as you make the sequence lengths longer. And here's concatenation. Okay? The same basic story with Astral. So if you don't bin, then you start off with a method that's worse than concatenation, and then it can become better. If you bin, it can stay at least as good throughout. So binning is improving species tree estimation. It actually, and this is really interesting, binning reduces the incidence of strongly supported false positive edges. So that's interesting because it's been thought that coalescent methods won't produce highly supported false positives, but they do. But binning reduces that incidence. Weighted statistical binning in general is benign to beneficial. It improves gene tree estimation, improves species tree topology estimation. It actually improves the branch length estimation, because coalescent analyses tend to make short branches when there's lots of heterogeneity. But if the heterogeneity is due to estimation error, you can actually, this can be wrong, this can be too short, and binning improves that. And it reduces the incidence of highly supported false positives. But it's not universally beneficial. There are cases that we have found where small numbers of taxa, strict molecular clock, and very high ILS. The combination of these three things can make binning a mistake. So if you are considering binning, don't do it if you have something like that. Small number of species, like at most 10, and average distance between true gene trees and true species trees, very high, and low bootstrap support for your gene trees. It can actually be a mistake. Here's an example of such a situation. This is very high ILS, the high ILS here, low ILS here. This is unbinned is in red, and then the threshold settings for binning affect things. This is what you get with Astral, this is what you get with NPS. And if you just look here, for example, unbinned versus binned, there's really no difference. With lower ILS, unbinned versus binned, binning can help, but it depends upon the threshold. With NPS, with very high ILS, you can actually make things worse. And the same thing is not true here, though. So with low ILS, it doesn't hurt. With high ILS, it can hurt. Okay, so now for the controversy. So Liang Lu and Scott Edwards published a technical comment in Science a few months ago, where they did two things. They actually tried to address the question of statistical consistency when you have gene tree estimation error. And the way they did that is they said, suppose you have a bounded sequence length per gene. And they said, we're going to try to prove that statistical binning, even in the weighted form, is inconsistent. So their proof fails for multiple reasons. But the easiest one to see is that their proof is actually using a theorem from Sebastian Rock and Mike Steele that only applies when you have an unpartitioned maximum likelihood analysis. Statistical binning always does the gene tree estimation error on every bin. It's using a fully partitioned maximum likelihood. So you can't actually use that theorem to analyze statistical binning. So their proof fails. That's one of the reasons it fails. They also did a simulation study. In other words, it's open. What they tried to prove may or may not be true, but they did not prove it. So the question remains open: what happens on finite sequence length? Okay, they also did a simulation study. That's a picture of their model tree. It's a strict molecular clock, a distant outgroup, very, very, very short branches with very high ILS. AD is 82%. That's an example of the kind of model species tree where things go badly. We could not reproduce their results. They had numbers that we did not get when we analyzed their data. We then got their data and we found the problem. And the problem is that they had two things. First of all, they had performed weighted statistical binning using unpartitioned maximum likelihood instead of partitioned. But even then we couldn't reproduce it because they had erroneous data in their supergene alignments. And what I mean by that, if you remember how statistical binning works, you take the gene sequence data, you then partition the genes. And so every bin is a collection of genes. And every bin should therefore have its data only from your gene sequence data. Some of their bins had data that could not come from any gene. So I don't know how it happened, but that caused huge error in the supergene trees that they computed, but does not affect gene trees, right? They use the original data for their gene trees, but they use this problematic data for their supergene trees. So that's their problem. But their simulation study is an example of exactly the kind of model that we have already discovered causes problems. So we reanalyze their data, we confirm that on their simulation, on the correct analysis of their simulated data, it does reduce accuracy. So yes, weighted statistical binning can reduce accuracy, but not by as much as they report. And with slightly larger data sets of the same model conditions, you don't see a reduction in accuracy. So it's really a thing of how many taxa, like under 10, and very high ILS just don't do statistical binning. Okay, so all the conditions that we have found where statistical binning hurts have these special properties. Okay, but they do ask a good question, which is, what are the statistical guarantees when your sequences are finite length, right? Suppose you only have 100 sites for every gene. Do you have statistical consistency? Can you prove inconsistency? That's a really good question. To date, there's only a little bit known. Sebastian Rock and I have a paper in Systematic Biology that says if you have a strict molecular clock, you can still do it, but only with some methods. If you don't have a clock, we have no idea. We really don't know if any method is statistically consistent. So we actually think that we can rephrase Liang, Liu and Scott Edwards' technical comment as a conjecture, which is to say they conjecture that for any positive integer L, if all the loci have at most L sites, then any pipeline that uses weighted statistical binning cannot be statistically consistent. And it's open. We do not know the answer. It's going to be hard to answer this. That's their conjecture, and it could be true. But there's no theoretical differences at this time between any of these methods. It's really just not known. This is published as well in PLOS Currents. So, future directions, better coalescent methods that are more robust to gene tree estimation error in particular. Better techniques for estimating gene trees, given that they're all co-evolving inside a species tree, or for co-estimating gene trees and species trees. And better theory about robustness to gene tree estimation error. We really don't know very much right now. And better single site methods like SNAP and SPD quartets, because you don't have to estimate species trees just by doing gene trees and combining them. The future might really be better addressed by having single site methods. Methods that don't go through gene trees, but just look at the patterns in the concatenated alignments. And with that, I just want to thank the people who did the work. Siavash Mirabag, who did the statistical binning, and Astral Bison, who helped with that. Bastien Brousseau, who was involved in our technical comment responses. And Sebastian Rock, who did a bunch of the theory. Thank you.
So we can take the lights. We have time for questions.
Yeah.
What do you do about recombination?
Okay, so if you have recombination within a locus, I think you really should look at shorter loci so that you don't have recombination, right? So we're theoretically assuming that there's no recombination within the loci. The impact of making your quote-unquote genes really short so that you avoid this is probably pretty bad, right? Really short sequences are not very conducive to good gene trees. So what I do, if I think there's recombination, is I would look at shorter sequences. But it's not clear how to estimate species trees when you have that situation. In other words, it's an open question. Yeah.
So, yeah, I had the same question. I was thinking about this paper that recently appeared by Gatesy and Mark Springer.
Right.
Kind of a crazy paper. But they were very concerned about this recombination issue, and I'm just curious if anybody's approached the problem from a simulation perspective to try to get a handle on.
The only paper I know of that has ever looked at this was with Lacey Knowles. I'm not aware of anyone having simulated in the presence of recombination beyond that paper. What we have done in some of our papers is look at what happens when you have very, very, very short sequences. So we have a collection of papers that have looked at species tree estimation when you have only 10 sites per gene. And and what's interesting is that it's not necessarily terrible. That you can actually, with enough genes, you can still do very well with some of these methods like Astral. So the only study I know that has looked at recombination explicitly is one by Lacey Knowles. Others have looked at what happens with short sequences. So those are the two things that people have looked at. And okay, the question is it really essential, so the same basic question, is it essential to keep your loci to the recombination-free part? Some argue it's not essential, and others would argue it is essential. I think that one argument that is made why it's not essential, if you have coding data, you're not going to have problems even with recombination. But what about using introns? The avian phylogeny was largely based upon introns. I don't know if you can, avoid these kinds of problems there. Yeah?
In the statistical pinning pipeline, the step from the coloring on the network diagram to the bins, is it clear that each coloring is unique so that the bins are
It's not. And we have, so what the question is, when you're doing statistical binning pipelines, you have the graph is defined by the gene trees and whatever bootstrap support threshold you're picking that suits your preferences. The graph is defined by that, but the coloring, there can be more than one coloring. And we've explored the impact of looking at multiple partitionings with different colorings. And we haven't seen differences that are significant. But what we have discovered is that if you do this and look at multiple different ways of doing the binning, and then look at what's supported in the different species trees you get back, you can get a better idea of what you have confidence in. So in fact, you could do this multiple times and look at different species trees as a result. Yes?
Yeah, you said when you have conflict among gene trees, you can't just pick the most common tree, but you can for four. Can you explain why that is?
So there's this, Noah Rosenberg, I think he coined it as the Anomaly Zone, which is when the most probable gene tree doesn't match the species tree. It's proven that for rooted 3-leaf gene trees, it doesn't happen. There's no anomaly zone. And for unrooted 4-leaf, it doesn't happen. But as soon as you go to rooted 4-leaf, it does. And as soon as you go to rooted, unrooted 5, it does. So, I mean, I could show you the proof, but it's basically, it's just a theoretical argument that you can have a problem with 5 and you cannot have a problem with 4. So, because you can't have a problem with 4, it's actually safe, if you only have 4 species, to just look at the most frequent gene tree on 4 species at a time. That's safe. It's just not safe on 5.
So there can be a problem, but it doesn't mean
On
it
5,
will be.
there can be a problem. It may not happen on your species. Some species trees have this anomaly zone. Some species trees do not. Right? So it's a property of the species tree. If there's low enough ILS, you're not going to get an anomaly zone. Yeah?
How does the statistical binning compare to like a partition maximum likelihood analysis?
I can't hear you. Can you?
How does the statistical binning compare to like a partition maximum likelihood analysis? So where you do like partition finder on as many partitions as you can, and you kind of
So the
like
question
binning.
is statistical binning compared to a partition maximum likelihood analysis. And partition maximum likelihood analysis can be extremely diverse or it can be not so diverse. We haven't, in terms of simulations, it looks like partition maximum likelihood is good if there's not too much ILS. In truth, the issue is really frequently just an ILS level issue. If there's not very much ILS, even unpartitioned maximum likelihood is fine. You know, fine meaning it's not bad. Not that it's consistent, but that it's not bad. So the question is, can you make things substantially better by doing partitioning? Maybe, and maybe not, right? That hasn't really been explored as far as I know. But if you have ILS, a lot of ILS, concatenation, whether it's partitioned or not partitioned, is not going to address the ILS issue. Because the partitioning isn't talking about, it's always assuming that there's one tree topology, and it's allowing branch lengths to change, it's allowing the GTR matrix to change, but it's not changing the tree topology. So these things that are really partition maximum likelihood, if you don't change the tree topology on the different loci, you're just not addressing the ILS issue. So if the ILS issue is large enough to cause a problem, that kind of partitioning doesn't help. It isn't addressing the real issue. Yeah.
Just based on your experience with analyzing lots of different empirical data sets, we don't have a simulation to start with. How prevalent do you really think ILS is?
Okay, I think ILS is prevalent, but that doesn't mean it's prevalent to the point where you need to worry about how you analyze it. I think that's really more the question. You know, it's got this AD level. I'm not sure where that is, but the average distance between, so it's further. The average distance between gene trees and species trees, you know, my guess is that it's not here. It's not here even for the avian study, right? The avian simulations are here, right? And the 1x means it's based upon what we saw in our estimated species tree. We figured that the average distance there was about 47%. That might be high, it might be low. But that's very far from this, right? So I think there is ILS. The question is there enough ILS to make a concatenation analysis inappropriate to the point where you would really want to use a coalescent analysis? A lot of data sets, I think not yet. On the other hand, we're at the beginning, as a community, we're at the beginning of method development in this area. You know, a year from now, 2 years from now, we may have coalescent-based methods that are really quite reliable to the point where you don't need to do concatenation anymore. You know, that could happen. And that's where we should be pushing. We should be saying, let's find out everything we can about where methods work well, where they don't work well, and come up with rigorous methods that have the statistical guarantees that we want and that perform well on data, right? But right now, I think, you know, if you have a data set and you're getting a difference between a concatenation analysis and a coalescent analysis, your judgment is going to have to come into play about what you think the right thing is. Right now, I think that we don't have necessarily the best methods.
I wish some journal editors had that same attitude.
It's not a religious response, right? You know, it's a data-driven response.
So it sounds like there's enough work to keep us all employed for a while. Thank you very much, Tandy.